Hey everyone, this is the last video for lecture, and like I said previously, you are not going to have a test for Chapter 8 itself. There wasn't one listed on your tentative course calendar because typically it's just hard getting all this in and then having enough time, lecture it all to you, get you all the information where you're able to fill out those study guides and start to study, and then give enough time for you to take a test on just this chapter and then turn around and study for the final and be able to take the final. And of course, with this whole COVID-19, it's made it even harder, but don't worry, guys. I'm going to work with you. So this is the last one. I'm giving you this information because, first of all, I think as a lab assistant, medical assistant, this is some very valuable information. And so what you can do is, this is your last videos, go over it, fill out those study guides that are in your binder, and then study this along with your study guides and information for both your lecture and lab final. I'm also going to be putting up some lab videos, and they're really just study material to help you study for your lab final and get you the information that you need. Okay, so we're going to move through these pretty quickly, but you can pause this and then you can follow along in your textbook. So we're looking at Chapter 8 on microbiology. So here's our learning objectives, and again, I'm not going to read each one of these out to you. They're also listed in your textbook, in Chapter 8 in your textbook, but you can read through these because the learning objectives are important, because at the end of this, at the end of me lecturing to you, which I wish I was doing in the classroom right now, I'm trying to get this information across to you, and the time in the classroom is time for you to raise your hand, ask questions, get clarification on things, and then also for you to be able to gather the information that we have found that is the information that's most important for you to know, and you studying those study guides, listening to the lecture, looking at the PowerPoint slides, reading the chapter, also the things you do in lab are things that you all put together to get as much information as you can. And these objectives are things that at the end of this, once you're through and you study, you should be able to do. So take the time to kind of just glance through and make sure you understand what's expected of you once you finish with this chapter. Okay, so I really hope that you have the textbook because it is a wealth of knowledge, and it not only will help you study for any certification exams that you will have to take as a laboratory assistant or a medical assistant, but it really will help you when you first go to work. It's something that you can go back and look at. It's also a great textbook if you're thinking about going back and going to nursing school or clinical laboratory science, or into respiratory therapy, radiology, any of these other medical fields. If you're wanting to continue your education, this textbook is really good. Not only does it have a lot of great information, it's easy to digest, but it also has a lot of really good pictures with a lot of procedures that show you step by step how to do it. Please utilize this textbook to help fill the gaps during this time when we're not able to be in that face-to-face class for both lecture and lab. Now, these are some review questions, and I'm going to move through them extremely quickly because I know this information, this is the last set of lecture slides, so I'm going to move through these quickly, but you may find them helpful when you're taking that last lecture test. So make sure you're pausing this and then trying to see if you're getting this right, because that last lecture test it up. These questions will be very helpful in helping you with that test. Okay, so starting in the overview of microbiology, and like I said, you can find this. It's in chapter 8 in your textbook, starting on page 202 and 203. You know, microbiology is the study of microorganisms, right? Including bacteria, fungi, protozoa, and viruses, right? Microorganisms are tiny, usually microscopic, which is can't see with that naked eye, right? Entity capable of carrying on living processes. Okay? And so, of course, if we're talking about tiny microscopic living things, then microbiology is the study of those things, right? So here we're talking about what is normal flora or normal biota, right? You have that normal flora that's in your nose, in your mouth, in your gut, both in your stomach and in your small intestines, and they need to be there, right? They serve a great purpose. They help us. They help us with our immunity. They help us digest our food, right? They do a lot of great jobs. But at the same time, they need to be where they are, for lack of a better word. In other words, the E. coli that is in your intestines that helps break down your food is doing a really great job there. But if it gets into your urinary tract, anywhere in your urinary, your bladder or your urethra, anything like that, then it can cause an infection. Okay, so again, I'm going to move through these really quickly because we're getting down near the end, and I'm really just wanting to get this information out to you guys because I know you're aware that we're getting down to the end, and some of you are graduating. Congratulations. And you need this information so that you can fill out your study guide so you can study it, because even though you're not having a test on the microbiology, you will need to know it for the final. And plus, this is really good information that you need to know. So right down there at the bottom, you'll notice there's a very good YouTube on your microbiome and how it works. You know, we talk about the lactobacillus is in the normal female vagina, right? That's where it goes. This organism creates a very acid environment. So certain, and it serves its purpose. It needs to be there. But then you can take certain antibiotics for something else. Say you got a upper respiratory infection or something. And so females can take these antibiotics, and because antibiotics kill bacteria, it can sometimes kill off that lactobacillus that needs to be in the vagina. And when it destroys that, then it messes up the balance, right? So then the Candida albicans, which is yeast, it finds that opportunistic time to take hold, and they go out of control and cause an infection. So not only do we need the bacteria to do its job, okay, we need it to be in the place it's supposed to be in, and we need to keep a delicate balance. So you will find some Candida albicans, some yeast in the vagina in very low amounts that's not causing any problem, but the minute the lactobacillus is not there and the internal environment changes from a very acidic environment to more of an alkaline environment, then it sets the stage for that yeast infection. So you guys make sure you read this, make sure you understand these highlighted words, because these could be on your final, right? And watch that video. Your microbiome is very important. I think a lot of us know about it, and we've studied it, we know what's going on, and a lot of us don't, and it's very important, and it's good to know that for not only your health, but when you're treating your patients. So what are pathogens, right? We're talking about microbiology, so we're talking about these pathogens, right? We're talking about viruses and bacteria, fungi, and parasites, right? So organisms that can cause disease, right? Only a small number of organisms, less than 1%, are pathogenic for human beings. So a person's immunity plays a large role in whether that pathogen will be able to produce disease or not. So healthcare workers must apply aseptic technique to prevent the spread of pathogens. That's why I go on and on when we're in lab about what do you do when you first come in. You need to wash your hands. You need to put your gloves on. then you need to get your 10% bleach and clean everything down, right? You need to make sure you're wearing your appropriate PPE. And when you're working in a clinic or a hospital, you want to be very spot on about all this, because we do not want to take that infection, go into one patient's room, and then take something that one patient has and take it into another room and spread that infection to another person, right? We talk about things like complex identification procedures of microorganisms are not typically performed by medical assistants or lab assistants. It's important to have, but having said that, it's really important to have a generalized knowledge of this, right? So what are some of the examples of microorganisms? Like I talked about, and there's really good detailed information in your textbook on page 203 about different kinds of viruses, bacteria, fungi, or parasites. So viruses, we're dealing with a lot of that right now, right, with COVID-19. So most of these viruses consist of either RNA or DNA, and they need to live in a host cell, right, and use that host cell's organelles to multiply, and they have to be grown on tissue cells, okay? So they need to hijack those cells and then use the cells' organelles themselves to be able to multiply. So I'm sure you learned this in your 100 level biology class when we talked about prokaryotes and bacteria and how they're classified. They're unicellular organisms that they don't have a true nucleus. And then they produce by binary fusion, and they form colonies. And these colonies are visible masses of bacteria found on a culture medium, okay? And then the culture is the reproduction of microorganisms in a predetermined laboratory culture medium under controlled laboratory conditions. It's just like when we take a swab of a throat, okay, and we put it on a blood agar plate, okay? Now what we're trying to do is we're trying to give this bacteria that is possibly growing in our patient's throat all the things that it needs to grow, so we can find out what it is and what antibiotic works best on it, right? So then we put that in that controlled situation of that incubator, because our body is at that same temperature, so that we can control everything to grow it out, right? So we have those prokaryotes that we talked about with they have a cell wall and a cell membrane, but no nucleus, right? Now that's the opposite of the eukaryote, which have no cell wall, but do have a cell membrane and a nucleus with a membrane. And there's your study tip down there for that. But I know you guys know most of this, that in your biology, your early 101s and 102 classes, you learned about these classifications of bacteria. So they're all single-celled organisms. We talked about how they reproduce with binary fusion, asexual reproduction. They can reproduce very rapidly. And I talked to you guys about those colonies, and if you see right there, every one of those little specks is a colony, and we put it on that media because that media has all the nutrients that bacteria needs to grow, along with making sure that you have it at the correct temperature. So now moving on, we talked about Candida albicans earlier, about yeast, fungi. They also grow on media, and there's different kinds of media out there depending on what you may think you're looking for. They're eukaryotes. They possess a true nucleus with a nucleus membrane. They lack chlorophyll, so of course they're not that green color, right? They're classified as either molds or yeast. So some fungi can take on both forms. The mold form being expressed at room temperature, and then the yeast form being at body temperature. Okay, so you can kind of imagine how that would work. Again, they're those single-celled organisms. They reproduce by budding. And if you look at that, and you'll hear people say budding yeast. And if you look at that picture up at the top, and you see the round circle with the little bud coming off, that's what they're talking about. It's like it pinches off, and then it pinches off and then comes a very separate cell. They form colonies as well, and they have visible masses on the culture medium. They have a very smooth, very creamy appearance. So if you look at that second slide right there over to your far right in the center, and you see all those little white creamy dots growing there, that's some kind of yeast there, okay? And that's been growing at body temperature, which would be in your incubator, right? The one right below it that has that real fuzzy appearance to it, that's more of a mold, and that would grow at room temperature. So in other words, if we set that plate out at room temperature, these molds could grow, that mycelium, you know, and when you look at them under the microscope, the yeast look like those little budding yeast under the microscope. But then if you look at that bottom photo over to your left, that's those kind of tube-like structures, and they have kind of a fuzzy appearance. That's how molds look under the microscope. And again, I know that you guys won't be looking under the microscope in your everyday job, spotting all the yeast and molds and things of that nature, but it's just really good when you're out there working to have a basic working knowledge of these things, that you can speak intelligently and professionally, and when other coworkers are talking around you, you'll have a real good idea of what they're talking about and how these processes work. Not to mention, if you further your education in healthcare, this is only going to be helpful to you. So here's one of our case studies. So take the time to pause this, get out a piece of paper, and see if you're getting the right answers. And again, guys, not only do y'all need to, you're taking everything you learn in lab and lecture from reading your textbooks and doing these slides and filling out your study guides, and you're putting it all together to be able to critically think through this stuff, so that when you're out there in the field, these are questions that you could think through and answer. So these are real helpful. You can go back and look at these and study these before your final. Okay, moving on to the last one, we're talking about parasites, right? Parasites. So these are stuff like hookworms, right? Giardia lambda, that protozoa that's up at the top. Okay, so the protozoa, like amoebas, right? And then there's other things like roundworm, flukes, and tapeworms. Parasitic organisms live in or on a host. They have to have a host. They need the nourishment from the host to be able to stay alive, okay? So Carl von Linné, a Swedish botanist, he created that nomenclature system for all living things, okay? And so, again, I know you guys covered this in your entry-level biology classes, and this is something that you need to know, especially how this is written out in the system. The genus is capitalized and the species is not, right? The genus and the species are always underlined or italicized. These are things, and if you look down here, you see Staphylococcus aureus, right? S. aureus, okay? These are just things that if you know how this works, that when you spot something, it'll be able to help you break it down. That's why MedTerm is such an important class. So here's another one of our thinking questions. So take the time to pause this and see if you're getting these questions right. Okay, so if you look on page 205 of your textbook, you'll see a really good picture of the different basic shapes that bacteria take on when they replicate. Okay, spirilla, which makes sense, is spirals, right? You see that in the picture. Bacilli is the rods. Cocci is the little spears, right? And then you have the coccobacillus, which is a combination of the spears and the rods, right? It looks like a cross between the two. Make sure you look on page 205 of your textbook, because there's some really good pictures on there, and it goes into talking to you all about the history of infection control, all the way from the 1300s down to 2006 when they were talking about the Zika virus. And every time we turn around, we have things like bird flu, swine flu, hepatitis, MRSA, MERS, HIV, Ebola, Zika, SARS, right? We know that as healthcare professionals, we need to be on the alert of these things. And I think sometimes when we go a long time without something really hitting hard in the U.S., we take for granted the things we need to do to protect ourselves, to protect our patients, to protect our patients' families, and frankly, to protect our families and our coworkers. So just make sure that you guys are reading the textbook as well. There's a lot of really great information in that textbook to go along with these PowerPoints. So you want to be able to describe each bacterial shape, okay? So a lot of these things speak for themselves, but make sure you take the time to understand it, because this is something you may see. So we got diplococci, which of course D pairs, right? And you'll see the little example of that. Then staphylococci, which is a grape-like cluster. And these are ways to help people, to help those microbiologists be able to identify what specific bacteria your patient has, right? That's why these culture and sensitivities are so important, and micro is so important, because you don't want to be giving antibiotics to a person who has a viral infection because you didn't bother to do a WBC count and see if it was possibly viral or bacterial, because a lot of these things mimic each other, right? And you also, do they have staph? Do they have strep? These are the patterns that they can look for, and these are signs that tells them what they need to, how they need to diagnose the patient, okay? Okay, so now, sorry about the dogs, guys. So now we want to talk about the most common pathogenic Staphylococcus. It's Staphylococcus aureus. It's a gram-positive cocci, and it forms those grape-like clusters that we talked about earlier. It causes the boils, pimples. It can cause abscess, wound infections. We mostly hear about people getting wound infections, and we know there's a lot of staph. It's just everywhere, and if we have an open wound, that staph can get into that open wound and cause a worse situation. So make sure you look through that and you guys are aware of this, okay? So then we talk about the most common streptococca, right? So, and then that Streptococcus pyogenes, right? It's a gram-positive cocci in chains, right? It causes strep throat. It also causes scarlet fever, rheumatic fever, pneumonia, infections after childbirth, all these kinds of things. And you'll see it's that little, you see the circled one? It's the cocci in chains, right? It forms chains. So when they look at this under the microscope and they see the little round coccyx in a grape-like pattern, they know that that's staph, along with that creamy appearance on this thing. There's things that they put together, plus little tests that they do with reagents that positively ID it as staph or strep, so that we'll know how to move forward with treating this bacterial infection. Of course, then we see the arrangement we talked about, Diplobacillus. It's arranged in pairs. You see this a lot in UTIs, which is really important, but there's other things like tetanus and gangrene, food poison, botulism, that kind of thing, okay? And then the spirilla, right? It's gently curved. You have that corkscrew or spring that can be a little bit different. So what's some of the diseases we're talking about with this? Syphilis is one of the main things. And if you see it down the bottom, see that almost looks like a spring? That's the syphilis bacteria. And then, of course, the cholera infection was more of one of these spirilla type organisms. So besides shape, what are other things about structure that you can find on the bacteria? So there you have the, again, I know that y'all have covered a lot of this in some other micro classes that you've taken and some other biology classes that you've taken, but make sure you understand about the how the phalangeal, the capsules, the spores, how all this stuff works, what it is, okay? So now we want to cover collecting, transporting, and processing microspecimens. Because what we want to do is first start, and again, you just want that basic knowledge of all those micro diseases and bacteria and parasites and fungi that you could run into. And then you want to make sure that you understand that when you're collecting it, when you're transporting it, when you're processing these specimens, some of the things that you need to do. So the first thing we always want to start with is safety precautions, right? That you should take when you're collecting some microbiology specimens. As always, we want to wear the appropriate PPE, right? And that changes depending on the situation we're in, what we're dealing with, and all that, right? So we want to make sure that we wear the appropriate PPE. You don't want to walk into a patient's room like you're on an episode of Contagion just to draw a patient's blood or to collect a minor specimen. But you also don't want to go in there and run the risk of, because you don't have goggles on or because you don't have a mask on, being able to have that infection go from the patient to you, right? Or when you're in surgery, you don't want anything, you're in that sterile area, you don't want a cough or something from you to get to the patient, right? That's why we have to select that appropriate PPE. We have to disinfect our countertops, our areas where we're doing collection. And I see, before COVID-19, I used to preach this all the time, and I would have people come in and say, well, Miss Shepherd, you tell us that all the time about cleaning your collection chair and cleaning this, but when I go, I never see them do it. Yeah, I understand that, and that's why stuff like that needs to stop. This is a prime example of why that needs to stop. We need to be disinfecting these countertops and collection chairs and all the areas in these exam rooms in between every single patient. You always want to make sure that your specimens are placed in transport bags after they're collected. They're sealed up properly, and you always want to wash your hands after any time you're dealing with a patient, right? So this is one of our thinking questions, but also I want to make sure that you look on page 206 and 207 of your textbook. It goes through collecting a throat specimen, collecting a nasal specimen, collecting a blood culture specimen, urine specimen. It goes through all the methods, and especially if you are a medical assistant, these are things I'm sure that you've been over before, but it's always good to go back over them to make sure. And if y'all notice, all those people that were out there trying to help be able to collect these specimens to test these people for COVID-19 that were doing those nasal swabs, you have to make sure you get down in there in the right place. So make sure you look at that, page 208 and 209, a lot of great information on collecting specimens, okay? So with a lot of great pictures and a lot of information that will help you during this time that we're not in the classroom. So make sure you pause your video now, read through your thinking questions, and see if you're putting this all together. Okay. So again, still talking about this, about the collecting, transport, and processing of your microbiology specimens, right? Okay, so we always want to try to collect our specimens before antibiotics are given and process them ASAP, right? You apply swab from the transport system, culturette, to the area, whether it's the throat, the back of the throat, eyes, rectal area, urogenitals, area of a wound. Then it's collected things are collected in containers, right? You have urine containers, blood culture bottles. You have other types of valves, collection devices that you can use to collect different types of body fluids, skin scrapings, abscesses, and things like that. You know, they may draw something off an abscess into a needle, and then it needs to be placed very safely into a container for it to be transported down to micro or sent to micro. You might have to send it to a reference lab so that they can culture all these things and see if there's anything growing in it. Okay, so here's how you collect a throat specimen, right? You want to take that patient, get them to tilt their head back, right? You use your tongue depressor, get them to say ah, which opens that movement of saying ah. You know, it makes the tongue go down in the back so that then you can take the swab, okay, and you rotate it circular in a figure or a figure eight, circular or figure eight pattern in the back. And what you're trying to do is, as you go in to touch that back of that throat back there, and as you're bringing that swab out, you want to try to avoid touching any other areas in that mouth, because like we talked about, you have bacteria and normal flora all in your mouth, and we want to make sure we get right back to that back area of their throat, so we make sure what they're going to culture is going to be where we think that infection possibly is. Okay, now we're talking about blood cultures, you know. When and how are blood cultures collected? Okay, so when the patient has some fever, it's usually a really high fever, and they're just not real sure where it's coming from, this is when they usually order blood cultures. The most important thing when collecting blood cultures is using aseptic technique, right, in the preparation of your puncture site, right? Most blood culture bottles require about 10 milliliters of blood in each one, aerobic and anaerobic bottles, and about 1 to 3 milliliters for one aerobic bottle for pediatric patients, okay? So remember, for adults, it's usually 10 mLs in each, and then for children, it's usually 1 to 3 mLs in just that aerobic bottle. And there's some information both on collecting that swab, which is on page 212 at the top, and it runs through everything you need. It's got a lot of really great information. To collecting your blood cultures is lined out on page 206 and 207 in your textbook. So here's one of those homework things where you have concepts and you describe things in your own words. So we want to talk about, in our own words, why is it so important for blood cultures to be collected aseptically? So you want to make sure your answer contains this. Okay, collecting those blood cultures aseptically means collecting it without any bacterial contamination at all, right? Because what we're looking for is the specific bacteria that's causing the infection, right? Not the bacteria that's normally just on your skin, right? It is so important because you do not want the doctor to think the patient has septicemia, which is often a life-threatening condition, unless they really do. So then, talking about urine collection, we've talked about this in lab before. So a clean catch, right? We went over this. A clean catch urine. That's when we give them the 3 different wipes, and we explain to them how they're to clean their genital area with the 3 soapy wipes, and then they're going to start to urinate some into the toilet, then move the urine cup into the stream, catch the middle part of the urine, then move it out and urinate the rest into the toilet, right? Then we tell them this because communication is key, right? To put that cap back on there very tightly, and then usually they place it in a window, or they bring that cup back to you in the lab and set it on the counter. So now we're talking about chlamydia. So what is chlamydia, and how is a specimen collected? It's the most common sexually transmitted disease, okay? And it's caused by chlamydia trichomonas, which is a tiny gram-negative intracellular bacteria that requires a host, as they do for cell growth, right? You may have no symptoms in men. In women, it can cause infertility. That's why we want to catch it early, okay? Again, you can turn in your textbook and find information on these as well, okay? So typically with this, physicians collect swabs from the endocervical canal of the woman or the urethra of a male and places it in transport media. If you notice that picture down at the bottom, that little vial there, they stick the swab down in there and it's set at a certain length to break off. And then you have a cap there. You'll see it, that little clear cap in their hand. Then they put the cap on it, and there's media in there to keep that organism alive until we can plate it out so we can make sure we find out what it is. We can also identify a lot of organisms by their DNA probe test, okay, to find out what their DNA is, so that way we can distinguish a specimen or organism from organism, right? Because a lot of these viruses, bacteria have a DNA profile or RNA profile that we can usually pinpoint to what they are. So then gonorrhea, okay? And I know a lot of you probably heard of this before. It's sexually transmitted disease, okay? And Neisseria gonorrhea, gram-negative intracellular diplococcus, looks like two kidney bean-shaped cocci facing each other. Women will have no symptoms or dysuria and a yellow discharge. Men have dysuria and a thick, creamy, whitish discharge from the penis. If untreated, it can cause infertility in both men and women. That's why we want people to be able to talk to us about this. This makes a lot of people uncomfortable talking to their patients about it, but we really want to talk to people about this, because if you can catch it early, treat it, get rid of it, then it doesn't cause these really serious problems of infertility. Same thing, physicians, during a pelvic exam, the physician will collect the swabs, right? And the specimen is swabbed onto a Gembeck plate with a small white pill. The pill, when activated, creates a 10% carbon dioxide atmosphere, which is needed for growth. The Gembeck plate, also called the Gembeck transport media, is then placed in a plastic bag and sealed up. Again, guys, it's like what we talked about earlier. What we want to do is give any of these viruses, bacteria, parasites, any of these things, funguses, we want to give them the atmosphere that they like to grow in. If we think this is gonorrhea, then when we're putting it in our plates, when we're doing our collection, we want to give them everything they need so they can grow. Because if that's what it is, we want to find out that's what it is, so that then we can treat it effectively, quickly. So here's another one of our thinking questions. Make sure you pause these, get out a piece of paper and see if you can answer these. This will let you know that you're reading, you're listening to the slides, filling out your study guides is actually working. And like I said, even though you're not going to have a test on this chapter, some of these questions may be on your final. Okay, and here's a case study. Okay, these case studies are important because it gives you scenarios that you may face as a medical assistant or lab assistant when you're out in the workforce, right? So this one talks about our patient named Ed Harris. He presented to his doctor with a white creamy discharge from his genitals. The doctor suspected gonorrhea. See, the doctor, we put all this stuff together, right? You guys know this. We put it all together like we're investigating a crime, right? So they come in, we get a history and physical. That's why we weigh them. That's why we take their temperature and their blood pressure. We ask them what their symptoms are. That's why when we look at this, then it gives us an idea. So we have an educated guess, right? A hypothesis. We think it's gonorrhea. So then what we need to do is take a swab from that area, and then we can grow it out, see if we were right, because we're not always right, right? If we are right, then we'll know exactly how to treat and monitor our patients. So what type of culture media that we discussed earlier should be used if we suspect gonorrhea? That Jim Beck transport media, right? And we talked about the special growth conditions that this type of bacteria requires to be able to grow. So how is this organism transmitted? Would his partner have necessarily known they were giving it to him? No. Often people have no symptoms. And if you saw on that earlier slide that 1 in every 4 person at some times during their lifetime is going to have a sexually transmitted disease. Okay? We've got a lot of stigmatism on this. People don't want to talk about it. They don't want to tell their doctors about it. They don't want to tell their physician's assistants, their nurses, their medical assistants that they're having this problem. We have to break down these walls. We have to stop placing these stigmatisms on these diseases. We have to stop taking our cultural biases and putting it in place and making people feel ashamed to talk to their healthcare professionals about their healthcare. Same thing with mental illness, with substance abuse. As long as we make people feel ashamed of sex and sexually transmitted diseases, as long as we make people feel ashamed of mental illness, as long as we make people ashamed of substance abuse, then they're going to be less likely to come into their healthcare provider and talk to them about it and look for a way to get better. What I want is this new young generation of lab assistants and phlebotomists and medical assistants and nurses. I want them to get out there and start tearing these walls down and start making people feel like they can come talk to their healthcare professionals about sexually transmitted diseases so we can start eradicating some of this stuff, especially in Caddo Parish. Okay, this is where you live. This is where you may work. If you live in Caddo or Bossier, there's high incidence of syphilis. And the only way we're going to stop this is by stop putting such a stigma on it so people won't be so ashamed to come and get treatment. So how would you document the culture for Neisseria gonorrhea? Remember we talked about charting, right? Date and time always, right? A culture for possible. You suspect it, right? But you do not know yet. So you always want to say that you are doing a culture for possible Neisseria gonorrhea was collected according to office procedures, your initials and credentials, right? So moving on, how should an ova and parasite, O&P, collection be made? Ova and parasite collection kits. They come with collection kits, depending on what you're looking for, pinworm, whatever it may be, with the preservatives that you need, such as formalin and polyvinyl alcohol, to keep this stuff preserved so that when we look at it under the microscope, we will see what it is, right? And the containers must be filled to the appropriate lines with whatever specimen you're collecting, whether it's feces or if it's a swab of some sort. And if you'll look in your textbook, there's a lot of good examples inside your textbook about, on page 208, there's a thing on collecting fecal specimens for ova and parasites and bacteria. It shows different types of collection kits that you can get. Also, on page 221, we talk about wet mounts, KOH preparation, pinworm specimen collection, and microscopic results. So make sure you look at that, because as a medical assistant, you could be setting up these wet mounts. You could be doing these KOH preps. You could be doing these pinworm specimen collection and microscopic results. If you haven't yet been able to do any of these, go on to YouTube when you get time. Look it up. There'll be a lot of great examples out there showing you exactly how to use it. Use your textbook. It goes through step by step telling you these collection procedures. And your white lab binder lays out a lot of this stuff for it. So guys, make sure you're taking the time to go through those labs that we didn't actually get to do hand in hand, but there's a lot of great information on there that tells you step by step what you need to do, from washing your hands all the way down to disposing of your trash, right? So when we talk about smears, stains, and wet mounts, right? After the microbiology specimen is collected, the next step, right, is that we need to identify what infectious organism we have. And a lot of times that means making wet mounts, making stains, making smears so that we can put it under the microscope so the doctor or a clinical laboratory scientist can look in there and see it. And as a medical assistant and a lab assistant, you might have the job of making these smears, of making these stains, of doing these wet mounts. So you need to know the steps it takes to do this. So guys, this is a really weird time with COVID-19. I know that it's really messed up everybody's ability to get as much hands-on as they want. But I always say to people, you know, you're going to get as much as you put in. If you want to be the best lab assistant you can be, the best medical assistant you can be, then take that time, guys. If these are things that I know some of you are in clinicals, some of you have clinicals you're going to go to, you know, take the time, you know, when things settle down for you a little bit, go back and look at these slides. I'm going to leave them up. Go on to YouTube, look for wet mounts and how it's done. Really take the time to help yourself learn all the things that you need to learn and fill those gaps in, and use your textbook as well. So we're talking about how bacterial smears are prepared, right? So a portion of the specimen is generally smeared onto a slide or mixed into a solution with water or saline on a slide. Like you have a drop of water or saline on there, then you take some of it and smear it on there, right? The specimen is then heat fixed, and if you look in your textbook, it shows you, it's a very good photo with examples of heat fixing. It's on page 212 down at the bottom, okay? So heat fixing, it denatures the protein and causes the swab material to stick to the slide, okay? Because we need it stuck to the slide, because when we start dipping it down in these stains so that we can see what's gram positive and what's gram negative and picking up the staining characteristics. Because just like when we stain our red cells and our white cells so that we can see which white cell it may be, whether it's an eosinophil or a basophil due to the stain it takes on, these bacteria are the same way. They absorb that stain, and it helps us see if this is a gram positive or gram negative, and it helps us identify exactly which bacteria it is. So making sure it's heat fixed to that slide is important, because if you don't do that, when you go to stick that slide down in the stain, it's just going to wash everything off, and when you look under there, there's going to be nothing. Okay, Dr. Hans Christian Graham developed the Graham stain more than 100 years ago. It's amazing to think it's been around for that long, isn't it? And we're still using it today. So the purpose of the Graham stain, like I talked about earlier, is just like our white cells and our red cells absorb certain stains and stain certain ways, so does our bacteria. So it's used to distinguish between the gram-positive and the gram-negative organisms. Gram-positive organisms appear blue and gram-negatives appear red. So what is the role of a medical assistant or a lab assistant in preparing a Graham stain? So although MAs and LAs cannot identify specific bacteria from a Graham stain, they should be able to prepare a stain slide, identify the bacteria as gram-positive or gram-negative, and give the shape and arrangement. So these are things you won't be doing it, making that final adjustment, but you should be able to prepare those slides, stain them, look, set that microscope up, get to the important place it needs to be, focus it in, look at it, and you should be able to determine if it's gram-negative, gram-positive, and what shape you're looking at. So we got possible sources, right, that we can get our smear from, right? A direct smear, right? You roll that swab directly onto the slide, right? Then there's a smear of one colony of bacteria growing on an agar dish, right, or agar plate. So you remove that one colony from the petri dish and rub that colony into a drop of saline or water onto a slide. And then smears are made from liquid media. You use a sterile swab dipped into the broth that is containing that media. That bacteria. That bacteria is growing in that broth, and then you roll it onto the slide. So those are three different ways. And there's a really good staining procedure that shows you how to stain stuff on page 213 in your textbook and 214. Then there's some really good talking about the KOH procedure and the wet mount procedure. So make sure you're checking your textbook, guys. A lot of great photos and information in there. So here's another one, our case study, a higher learning activity. So make sure that you pause this video and be able to see if you're putting all this together. So here's some smear preparations going through that. There's some pictures here that kind of show you. Then there's also some stuff in your textbook. So make sure you pause this and go through it and understand about the crystal violet, which is purple. You know, your iodine, which is your trapping agent, right? Alcohol or acetone, which decolorizes. And then the saffron stain, which your gram positive is purple and your gram negative is red. So what's the most critical step in your gram stain procedure? It's the decolorization step, right? Because if you saw on that slide before, all of them take on that blue, right? And so if you don't decolorize, if you miss that step, then when you look under it, everything's going to be purple, okay? If you decolorize too much, everything's going to be red. So you want to make sure that you follow that procedure step by step exactly like it's supposed to be. And a lot of times these come in kits, it's going to tell you exactly how long to leave on everything. The main job is that you follow the procedure to a T. So what causes the bacteria to take on the purple or red color for the gram stain? Okay? So we want to understand about gram positive. They hold on to that crystal violet because of the large peptoglycan layer that's in their cell wall. Because it's so large, they will hold on to that, so that when you go in there and try to decolorize it, it doesn't decolorize as well. So the gram positive usually holds on to that crystal violet. But the gram negative organisms have a thick outer layer of lipoprotein and lipopolysaccharides, which are dissolved by that decolorizer, okay? So that goes away. So now that outer layer is gone after you decolorize. This allows that crystal violet to be washed away by the cell rinse, and then the gram negative organisms therefore are colorless after they're decolorized, and then when you apply that red saffron, then they take on that color. So quality control, right? We always got to run quality control. And the way we do this is we take a known, okay, that's your key word, known. A known gram negative and a known gram positive organisms are stained along with the patient specimen to make sure the colors are correct. So you do your known gram negative, your known gram positive, and your patient all at the same time. You do them all the same way, same person doing it, okay? So that way, if your known gram negative shows up like it's supposed to, your known gram positive shows up like it's supposed to, then it's a better way to trust that your patient is correct. So how are those test results reported out, right, for a gram stain by the MA or the LA? Okay, you state the color as gram positive, purple, or gram negative, red, the shape and the arrangement. Okay, describe the organism in picture A. So I'm going to go through these quickly. I'm just going to sit here because I want to get this information out to you guys as fast as I can, and I want to make it as short as possible so you can go through it, get your study guides filled out so you can study. And like I said, there won't be a specific test on Chapter 8, but some questions may be on your final because I want y'all to be able to move on, especially graduates that are graduating this semester, move on to start studying your study guide for your final, okay? At the same time, these videos will be up all during the summer. I hope you have your textbook. I hope you keep your textbook because you can go back and study more of this. Here's another one of our case studies. Again, guys, pause this and see if you're getting the correct answers. So now we're moving on to acid-fast smears. Okay, so what are acid-fast smears used for, right? Well, when we talk about Microbacterium genus, those acid-fast bacilli, that's what they're referred to as, when the bacteria appears red with that acid-fast smear. So what you're looking for is this all boils down to, guys, to you being able to properly identify what bacterium is growing in your patient, right? We want to positively ID this stuff. And these are ways with these stainings and the stain that the bacteria takes on, the different types of shapes that they are all tools that we can use to help us be able to identify which bacteria our person has, right? So tuberculosis, right? Okay, so we've all heard of TB. We get our TB skin test done, right, to make sure that we don't have TB, right? So an infection in which tuberculosis, which is nodules, are formed in the lungs and is caused by that microbacterium, right, tuberculosis. And just like we talked about that up above, we're talking about that genus, Microbacterium, right? There's different kinds. This tuberculosis would be an acid-fast bacilli, right? So if we stain it, it would appear, the bacteria would appear red, and that's how we would know it would be in there, right? Some other symptoms for TB include those night sweats, bleeding in the lungs, they'll spit up some blood, and having that purulent sputum. These are all symptoms of it, along with, it's like I talked about before, we take all these things and put them together to be able to positively ID our patients and what's wrong with them. So we will make sure that we'll be able to effectively diagnose. So again, that Mycobacterium is the genus, right? And then we have tuberculosis. We also have this avium complex, MAC, right? MAC. It's also an acid-fast bacilli. But it usually ends up being something that goes through the bodies in an AIDS patient, right? Both TB and MAC have been on the increase as the result of AIDS patients, because as we know from talking about our last chapter when we were talking about our immune system and our T cells and how our immunity works, with AIDS patients, it decimates their immune system, so it's so far down they have no immunity, so they are really, really susceptible to things like TB and MAC. So especially in this picture down the bottom is most likely taken from an African country, AIDS is decimating that country. And so when you talk about people, when that AIDS goes up, then also all these other diseases go up as well, because the AIDS patient's immunity is down and they come up with all these other diseases like TB and MAC that kill them. And what we want to learn is all the different things, all these different ways in which the tools and tricks and tips and things that we have to be able to positively ID our patients, right? So we're talking about two methods of performing an acid-fast stain. The Zeele-Nielsen method, you use heat and the Kenyon cold stain mixes a detergent with the dye to break down the wax in the cell wall, okay? So what you're trying to do is break down that cell wall. It's like we talked about earlier, because what you need to do is once you break down the cell wall, then we use something, right? Some type of solution. We talked about earlier about the saffron red, and we talked about this carbofusion solution as the primary stain. So once you decolorize with the acid alcohol, then you counterstain with the methylene blue. Okay, so same type of thing. You're doing something to break down that cell wall, right? You do initial stain, break down the cell wall. Once you break the cell wall down, then you decolorize it and get all that initial stain out of there, and then you counterstain with the methylene blue. So anything that is in there, okay, that is able to be, we're able to break that cell wall down, then it allows that methylene blue to get in there so it stains it, right? So again, we want to go through these thinking slides so we can take what we go over and make sure we're learning it. So guys, really take the time to pause these. You can fast forward through what I'm talking about, but take the time to pause these and really see if you're putting all this together. So we talked about wet mounts, and like I said, there's some really good information inside your cell, and this is a big thing to know about because a lot of times as a medical assistant, you really will probably have to set up a wet mount for one of your physicians or look at a wet mount. On page 221 of your text, it talks about how wet mounts are rapid microscopic techniques that are done to view organisms in their living state, right? Because we want to see how they move. So we want to see their mobility because seeing their mobility helps us tell which one it is. And this one is a drawn out version of Trichomonas vaginalis. So we have different types of wet mounts, right? So what the different things that we use wet mounts for? So what are the examples of certain type of wet mount to use for? So we use them to detect the protozoa, such as Trichomonas vaginalis, which is a very common sexually transmitted disease. It produces itching, a frothy, creamy discharge. Trichomonas has a very characteristic jerky movement. As a matter of fact, there's been many a times when I would set up a wet mount and I would go as a lab tech and I would go to look down in the microscope and they would be moving around so much it would almost feel like they were jumping out at me and I would back up. So make sure that you copy this link, go to this link, look at this video so you can see what these trichomonas really look like. So to detect the presence, I'm sorry guys, I'm getting tired. It's late in the day. I'm working till all day today at 6:30 at night to get these things done so I can get them up as soon as possible so you guys will have all the information you need to start studying for your final in lab and lecture. So forgive me if I'm stumbling over my words more than normal, okay? So to detect the presence of clue cells, okay, these are vaginal epithelial cells that are covered with Gardnerella vaginalis, which is a gram-negative coccobacilli found in women with bacterial vaginosis. Infections produce a grayish discharge and a fishy smell in the genital area. Sorry about the dogs, guys. So again, get this YouTube video and go and look at it. It'll give you a really good idea of what these things look like. Okay, here's another one of our case studies. So take the time to pause this video, read the case study, get out a piece of paper and see if you can write this down. And I know this is hard, guys. These PowerPoint videos were set up to be in a classroom in a face-to-face setting, okay? And due to the emergency nature of what's going on, this class was not really meant to be online. So I apologize for the information, but the main thing I'm wanting to do is just make you be able to take the things that you're learning in lab and lecture when we actually were in face-to-face classes, and then take these PowerPoints with the information that's on there, take those study guides that you have, okay? Fill them out, read your chapters, and put all that information together to be able to put it together and be able to do these case studies so that if you're met with this type of thing out in the real world, you will be able to critically think through it and be able to take care of your patients, okay? Guys, make sure you pause these and really go through this, because this is the information that you need to know. Not only do you need to know this for when you're out there working, but for any certification exams you're taking, and there will be questions on your final. So make sure you pause this and really go through this, okay? And then get out your study guide for your finals and start studying those study guides for your finals. So now we're still talking about that KOH prep, okay? We're talking about that KOH prep, which is potassium hydroxide, right? And the KOH prep is used to identify fungal infections, things like Candida albicans that we talked about earlier, right? So remember, like I told you, on page 211 of your textbook, there's some really good information on the wet mounts. There's some really good information on your KOH prep. These KOH preps is just another type of microbiology slide identification, right? Okay, the KOH is a strong alkaline solution that breaks down protein material and clears the tissues. This allows any funguses that might be present in the skin scrapings or vaginal specimens or mucus to be seen under a microscope. It's just like breaking down. What you're doing is prepping these things. You're prepping this bacteria. You're prepping these funguses by tearing down the walls, or by prepping them in a way like here with this alkaline solution, so that you will be able to see them under the microscope, okay? We want to get everything else away and just highlight, if it's there, these yeast and molds and fungus, right? And you want to make sure that the KOH preparation procedure check sheet is in your workbook, okay? And is demonstrated in procedure 8.4, located at the end of this section. So make sure you go through that. Make sure you go to those worksheets, and that you know how to demonstrate step by step how this is done. So the KOH, when mixed with the specimen on the slide and heated, you see it right there over that flame, and then alkaline solution breaks down that protein material such as hair and mucus and stuff to clear it all away, so it helps facilitate the identification of any funguses that might be present in these specimens. So here's another one of our case studies. So do the same as always. Pause them, get out your piece of paper, and see if you're putting this all together. So now we also, on page 211 down at the bottom, there is a figure 8.13 that shows you how using this cellulose tape procedure for collecting pinworm eggs, okay? So we talk about that genus for the species name for the pinworm, Entomebus vermicularis, right? Entomebus vermicularis. This is the genus and species name for what we commonly refer to as pinworm. And here's some pictures of the parasite and the ova and parasite, right? So talk about the life cycle of pinworm, and we need to understand what the life cycle is when we're collecting this stuff. So the female pinworm, she lays eggs in the anal area of the host. I know it's disgusting, right? But this is what happens. So commonly a child, right? So the process causes itching. So the host scratches the area, infecting the hands and fingernails, okay, with worm eggs. Then the host puts their hands in their mouth or the mouth of family members, and the eggs are swollen and mature in the intestinal tract. I know this all sounds disgusting, but it's important to know. You'll see this a lot, and you need to know how to properly collect these specimens. This is why washing our hands, teaching our children from the earliest age to wash their hands. Washing your hands, if you teach those kids to wash their hands from an early age, they will know that this is something they should do through life. So make sure you look on page 211. It talks about Entomebus vermicularis and how it's this tiny, thin, and we saw that in that picture, white, round worm, commonly known as a pinworm, right? Okay, so, and then the procedure for collecting that is down at the bottom, okay? So watch that video, because these are all ways. I know we're not able to get in those labs and literally put our hands on things, but if you look at these photos and go through these procedures that are in your textbook and go through the procedures that's in your workbook, if you watch these, YouTube videos that I'm providing for you so that you can actually see this stuff. I know it doesn't fully take the hands-on thing, but it gives y'all a lot of information to put together to be able to know how to do these things. And a lot of these things are CLIA waived, right? So they're procedures that are not really highly complex. So if you do all this stuff, if you read the book, if you watch the videos, if you watch these PowerPoint presentations, if you go through your workbook and you do these things step by step, you will have the tools you need to perform those out in the field. So again, here's that procedure. Again, here's some good example of that cellulose tape and how it's done. We talked about earlier. And like I said, there's also an example of this on page 211 in your textbook. So make sure y'all go through this, watch that video as well, okay? So still talking about pinworms, the MAs or LAs, they collect the specimens. You need to know how to do this because you may be put in a position to collect this specimen. Then they prepare it and focus the slide under the microscope. Then a physician or clinical, some type of MLT, MT, a clinical laboratory specialist will come in and examine that tape that is on that slide to look for those Intermebus formicularis eggs, which you see down there at the bottom. You'll see that picture of one of those eggs. That's what it looks like. So over to your right-hand side, down at the bottom, that's the picture of the egg and what it would look like under the microscope. Right above that is someone collecting that specimen, okay? And above that is the tape that is on that slide, right? Or on the, I'm sorry, the first one closest to you is on the, like a tongue depressor. And that's what you use to touch the inside of the anal area of the child. Then you take that tape and move it from the tongue depressor onto a slide, then put it on there and focus it in, right? So here's another one of our case studies. So now here we're still talking about CLIA waive test for Streptococcus group A testing, right? Okay, so some Streptococca are capable of producing hemolytic toxins, okay, which hemolyze RBCs when grown on sheep blood agar. So if you look at that top, you can tell right there how it has just hemolyzed that blood, and now instead of it being red, it's white. So if you look at those pictures, that shows you, and that's one of the tools that a microbiologist or clinical laboratory scientist or physician would use to help identify this Streptococcus, okay? Okay, guys, I'm going to stop right here because I want to get these uploaded onto YouTube so that y'all can get the first part of Chapter 8. And then, starting bright and early Friday morning, I'm going to quickly go through the rest of the slides dealing with Chapter 8, and then I will upload those as well. But this will get something out there so that y'all, once you get through with Chapter 5's test, then you can move on to worrying about make sure you know this chapter on microbiology, Chapter 8, and then I'll put the rest up on Friday. We can get this out of the way so we can start focusing in on the finals. So hang in there, guys. If you have any questions, comments, or concerns, make sure you email me, and I will see you on the next video.